come to our next speaker, Dr. Rima Jabber from Evonik. Dr. Rima is the Global Product Manager, Lipids Business Line Healthcare at Evonik. Dr. Rima joined Evonik in 2009 and has held multiple R&D roles in Germany and abroad. She has over 10 years of experience in parenteral and complex oral formulations and the handling of highly potent active pharmaceutical ingredients. In her previous positions, she also gained experience in managing the development of new biomaterials for medical device applications, managing publicly funded projects such as BMBS, EU and Australian linkage grants, and new manufacturing methods, biotransformation, and plant cell fermentation. Since 2022, Dr. Rima has been responsible for the global product management of lipids at Evonik's healthcare business line. Dr. Rima is a pharmaceutical chemical engineer. She obtained her PhD from the University of Jena in Germany, where she specialized in optimizing processes for biodegradable polymers, including for manufacturing and formulation. Dr. Rima is the author of several research papers and patent applications. So we welcome you, Dr. Rima, and look forward to hearing from you, about mRNA. I think Dr. Vivek Jha also was mentioning in his presentation, in his talk, that this is something we should look into with the COVID era that has just passed us. So it'll be good to listen to you, and over to you, ma'am.
Thank you so much. Thank you for having me. Just give me a second. I will share my slides. Maybe you can give me the...
Yeah. They'll be just giving you the control. Yeah, you have the control.
Just give me a second. Can you see my slides?
Yes, I can see it.
Okay, perfect. And full frame?
Yes, full screen. Yes.
That's good. All right, so thank you for having me today. It is my great pleasure to represent Evonik Healthcare and to talk to you about the role of lipids in the delivery of messenger RNA using lipid nanoparticles. I have organized my speech into, or my talk into a couple of points. I will be introducing Evonik briefly, discuss a bit about the opportunity we have with the messenger RNA technology that would revolutionize and change the pharmaceutical industry. I will also walk you through the different compositions of the lipid nanoparticles and the role of the functional lipids and how do they play a major role in delivering the API. I will also present a case study comparing the vegetal-derived cholesterol versus animal-derived cholesterol. And I would like to show as well how Evonik contributes to the value chain of messenger RNA, LNP therapeutics, and our role to address, and how did we address the pandemic. So Evonik, who are we? We are the healthcare branch of Evonik. Evonik is a specialty chemicals company that is based in Germany with operations worldwide. We are an innovation hub for world's leading companies in pharmaceuticals, nutraceuticals, and the medical device market. Evonik is one of the leading CDMOs for complex parenterals, including lipid and polymer-based drug delivery. We also offer within Evonik different kind of services that is needed for clinical development. Besides nucleic acid, we also work with the lipids formulation for small molecules, peptides, and proteins, including high-potent APIs. We support our customers throughout the whole drug development cycle. This includes backward integration into the, let's say, backward integration into the key excipients such as polymers, PEGs, lipids, and custom lipids, and development of formulation services from commercial up to the fill finish. And here, Dr. Jaha have mentioned how the market is developing. So in the past, drug research in the pharma industry was all about chemically synthesized small molecules. We had to test millions of compounds just to find and identify the right API for a certain disease. The market also shifted in the last 20 years. So currently we are moving more to biologicals. So we have recombinant proteins and antibodies, which is biologically produced outside of the body. And almost 10 or to 20% of the market right now is based on biologicals. However, it is a drug and it is future, it has shifted the category of messenger RNA. And you have an for individualized medicine. And it's, let's say, the application for it is huge, and we believe that it will change as well how we tackle new diseases, or let's say oncology diseases, and hopefully not new pandemics, but also for any possible pandemics where we can bring vaccines faster to the market and bring therapeutics faster to the market. So how do we deliver these messenger RNA therapeutics and how, or the nucleic acids? We usually apply lipid nanoparticles, or we use lipid nanoparticles. So what is a lipid nanoparticle? A lipid nanoparticle is basically the cargo and a lipid that is protecting this cargo. And the lipid nanoparticle, let's say, each component of these lipids plays a critical role in manufacturing and formation of these particles. We start with the nucleic acid, which is in the right in the middle. It is our active payload that we need to keep intact, and we need to make sure that it arrives safely to the body. For the lipid components, we have ionizable cationic lipids, we have PEG lipids, and we have cholesterol. All these functional lipids I will be covering in my next slide. As for the structural lipids, or we call them sometimes phospholipids, such as DSPC or the dopey, which makes around roughly 10% of the lipid nanoparticle composition, they have an important role of making sure that the stability of the lipid nanoparticle is guaranteed. So they guarantee, or they make sure that the stabilization of the lipid nanoparticle during the manufacturing and the long-term storage happens. Moving to the ionizable cationic lipids. The ionizable cationic lipids makes, let's say, almost 50% of the lipid component in the LNP, and the role of the ionizable cationic lipids starts at the very, very beginning of the lipid nanoparticle formation. So the messenger RNA is usually prepared in an acidic buffer, and we usually dissolve our lipids in an organic solvent. They get mixed together, and then you have an acidic environment. Now, our cationic lipids have usually a pK between 6.5, 6.3 to 6.9, and with the acidic environment, they get completely protonated, and therefore there is an electrostatic interaction that happens between our nucleic acid, or our messenger RNA, and the cationic lipid, and they build a complex together. So the deprotonated again, and the payload then is released. And that shows that the ionizable cationic lipids have a very important impact on the potency. The role of the PEG lipids. The PEG lipids, they constitute the smallest molar percentage compared to the other lipids. They make around 1 to 0.5% of the whole composition. And they influence several key properties such as the nucleic acid encapsulation efficiency, the circulation half-life, the in vivo distribution, and the transfection efficiency. Now, all these properties are somewhat related to the molar ratio of the PEG lipid, as well as the structure of the links between the PEG chain and the lipid chain. And one of the other roles, or the main roles that the PEG lipids play, is they control the particle size of the LNP, especially when they contribute in the LNP self-assembly. So we have this citric barrier that, let's say, the hydrophilic citric barrier that the PEG chains form at the LNP surface. And the citric barrier also supports the LNP, or supports the particle, to prevent it from aggregation. So it ensures the particle stability during the preparation of the particle and during the storage. Cholesterol. Now, the cholesterol constitutes around 38% of the lipid components in the LNP, and it has a significant role on the stability and the overall encapsulation of the payload. And there are two types of cholesterol that are in the market. We have the animal-derived cholesterol, which comes from the sheep wool, and we have the plant-derived cholesterol, or the vegetal-derived cholesterol. In Evonik, we have our own brand, which is called Phytochol Inject, and it's an alternative to animal-derived cholesterol. It has a high purity and a consistent quality. We reduce the risk of contamination with our plant-derived cholesterol. We don't have a batch-to-batch variation, and we meet the USP, the European Pharmacopoeia, and the Japanese Pharmacopoeia. One more point that we have as well, that we of the new effect in January to see nice. And we have a lot of and we just secure the supplier for Phytochol. Can you still hear me?
I can still hear you. Why don't you put your video in off mode? I think you can just stop the video, so then we can hear it. You can keep your video in off mode.
Okay, just give me a second. Is it off right now?
Yes, the video is off. I can see the background, yeah, and I can hear you. I can hear you, so you can continue with the presentation. I can't see the presentation right now.
Yeah, maybe you can share also the slides, because I think it just went down. I don't have it.
Okay, fine.
Yeah.
We can hear you. Yeah. So, Karthik, could you please run the slides?
Yes, Shivani, I will share now.
Yeah.
So what was the last slide that you saw from me?
It was about Phytochol Inject.
The Phytochol. Okay.
Phytochol Inject. Yeah. That was the last slide.
Are the slides being shared? Not yet, right?
I think he's doing it. Karthik, are you sharing the slides? Yes, he's sharing the slides.
Okay.
You can, yeah, next slide.
Maybe you go to slide 10?
Yeah, behind that.
Okay, perfect. Okay.
Yeah.
So that was the next slide on the phytophil that I wanted to show. So there are currently 20 products in the market that are based on lipid-based particle technology. It varies between liposomes and lipid nanoparticles, and almost 75% out of these products, they contain cholesterol. Of course, some of them are using animal-derived cholesterol, and some of them are using the vegetal-derived synthetic cholesterol. And that shows as well that there is no huge difference between both of them. But if you go to the next slide, please. We have conducted a case study to compare the vegetal-derived synthetic cholesterol compared to the animal-derived GMP materials. And what we wanted to see if there will be a difference in the product characteristics and during the manufacturing. Next slide, please. So what we have applied here for our study, we used the poly-A, which is a surrogate of messenger RNA as an active ingredient. And we applied or we manufactured 3 sets of batches of lipid nanoparticles that were manufactured using a tea mixing and using the 2 types of cholesterol, the animal-derived and the vegetal-derived, our own phytochol. So the lipid composition or the formulation, we use the standard one. We have applied a TSPC, the phospholipids, the cholesterol, a cationic lipid, and the PEG lipid. Next slide. Thank you. So for the manufacturing of the nucleic acid lipid nanoparticles, usually you go into an upstream and a downstream process. Now the upstream process consists of a single step of a rapid mixing. So as I explained before, we dissolve the lipids in an organic solvent, the poly-A or the messenger RNA surrogate. We dissolve it prepared it in an acidic buffer at a low pH. Now, after the mixing, we go into the filtration, the tangential flow filtration, and this is where the exchange of buffers takes place, and we move it more to a neutral pH. We go after that to clarifying filtration and to a sterile filtration, and we store our product at -80 Celsius because, or due to the limited shelf life that we have, if we store the particles at 5 Celsius or -20. Next slide, please. What we looked at or what we have measured was the drug content and the encapsulation efficiency through the different steps of the manufacturing. So post the tea mixing, the TFF, the tangential flow filtration, where we exchanged the buffer and the pH, and also post the clarifying filtration and the sterile filtration. And it was an important step to look at these parameters here due to the shear forces, and we don't know exactly if there would be a difference when applying a different kind of cholesterol, a vegetal cholesterol or phytochol. What we also did is that we measured the drug content and the encapsulation efficiency post-freezing thaw, because we measured, you have to measure your particle and to see if they are still as stable and if still have the same drug content and the encapsulation efficiency was not affected after the thawing. So we did a 1 cycle of thawing, and the results showed that there was no difference between the animal-derived and our phytochol cholesterol. So at all stages of manufacturing and after, or let's say post-thawing, there was no difference observed in the poly-A content percentage or the encapsulation efficiency. Next slide, please. Yeah, so we also tested the physiochemical attributes of our particles. We looked at the theta potential, the polydispersity index, the pH, and the osmolality. And we also could prove that there was no difference observed in the physical characterization between animal-derived and the non-animal-derived cholesterol. Next slide. So as a conclusion, we can say that lipids prepared with animal-derived cholesterol and phytocol showed similar in-process characteristics and final product characteristics, and substituting the animal-derived cholesterol have a minimal impact on the manufacturing and the physiochemical attributes. Next slide. So in the last slides, I have talked a lot about the role and the importance of the lipids in the LNPs and in the formulations, and how critical in enabling, how critical they are in enabling the vaccine or the therapeutics to function. But we need to take a step back and just to mention the importance of the quality and the purity of these lipids, because having impurities in our lipids can affect the behavior of the therapeutic and affecting the release of messenger RNA. There have been a couple of studies that have been conducted that showed that some kind of impurities in the cationic ionizable lipid can influence the messenger RNA release and can influence as well the behavior of the vaccine. So Evonik is let's say, a company that has been only 2 years in the lipids field, where we manufacture lipids and we bring it to the market. But the messenger RNA vaccines, they have been also only for 2 years so far. However, we have been in the market for almost 30 years through our Vancouver labs, where we have years of formulation experience and formulation development. We worked with almost 90% of the companies who are active right now in the messenger RNA or the nucleic acid field. And we know firsthand that the quality of lipids can play a considerable role and an important role in the therapeutics of vaccines, and the vaccines, sorry. So Evonik Healthcare, at the moment, we have off-the-shelf products, our phytocol, but we also support our customers with custom lipids, with ionizable lipids, PEG lipids, and we support our customers as well in the pharmaceutical development of formulations and clinical trial material, as well until commercial, as I have mentioned before. So we contribute to the value chain from the very, very beginning until the commercialization of the product. Next slide, please. So our role in the pandemic. Evonik supplied at least 3 critical lipids in significant quantities for the COVID-19 vaccines. We are very proud of this achievement. We were able to bring up these lipids to the market in less than 3 months. And our contribution, we made a key contribution to the COVID-19 vaccine availability to the market. And that didn't come out of a coincidence. Evonik has a deep technology understanding and a deep know-how when it comes to our process development, optimization, and isolation of products. Where we have, this is our bread and butter as well, our backward integrations of fats and PEGs and other raw materials. Next slide, please. Yeah. So we have noticed that, or that's the trend that we're seeing as well in the market, that a lot of customers are moving into proprietary lipids, especially for ionizable or the MPEGs or the PEG lipids, and even the messenger RNA vaccines that are at the moment in the market. They both have proprietary lipids for cationic lipids. And we can support our customers where we can support them from the very earlier beginning of lab formalization, where we develop the process for them, optimize it, support them with non-GMP samples until commercial manufacturing. Next slide. And that's my last slide. So the future, how does the future look like, and where are we heading? So we're expanding. We have signed an agreement with the US government of a $220 million US to build a global scale lipid production facility to support our customers to develop nucleic acid drugs with lipids and supply them through commercialization. We're expanding as well in Germany. We are building our lipid launch facility for clinical development. It's supposed to be online and or active in the beginning of next year, Q1, and where we support our customers with smaller batch sizes for GMP lipids. And with that said, I'm done with my presentation. That was my last slide. Thank you so much, and excuse the problem with the video.
Right. Thank you, Dr. Jabber. And just a short recap of your presentation, for the audience plus our understanding. So during the COVID times, we heard a lot about, lipid nanoparticles and mRNA. Thank you for explaining the technology and, it was very interesting to understand what are we talking about, because these are buzzwords we heard a lot, but now I think you get a sense of what it is exactly. And like how you mentioned, vaccines and therapeutics, people are looking for, getting faster to the market. Definitely, this is an area where a lot of work and needs to be done, So Evonik can make a contribution to this. We look forward to that. And one more thing you mentioned was, research and commercialization to the market. And you mentioned about your lipid portfolio. It is very interesting to understand that we see the final vaccines or the medicines that are helping us, but what has gone into it, it is something you brought to light. That was very interesting from say a consumer perspective and from the industry perspective. And last but not the least, I'd like to thank you for waking up so early in the morning, joining us so early in the morning from Germany. And we are enjoying the mid-morning afternoon time in India, but it's really early for you. Thank you so much for your patience, and joining us so early.
You're very welcome. Just to recap on what you have mentioned, that the nucleic acid or the new biologicals, they are all based on the same principle. So it is faster to market. So it's the same principle, you manufacture it the same way. It is the messenger RNA or the nucleic acid, usually the development of it until to bring it to the market does not take as long as a standard biological or let's say a small molecule. So it is faster, we can develop it in a faster way. Of course, the lipids have to be the proper one. They have to have the high quality that we have mentioned. And we rely, at least in Evonik, we have, we make a lot of, or make a big deal out of the quality of our lipids. And it's a very important aspect because you can ruin the whole formulation out of the bad quality of a lipid.
Thank you, and we'll look forward at the Q&A at the end of the session. Right?
Thank you.